Thank you very much. And we heard a lot about adjuvants, and I will continue talking onto this. And so now I hope everybody's hearing me. And as I'm as well an assistant as a professor at Case Western Reserve University, as well having multiple hats as many other speakers as well had, I'm from Cellular Technology Limited, and I need to disclose this. And shortly, our company has 3 departments. One is the contract laboratory, where we perform work for our clients, then the instrumentation, where we actually do the analysis of the data by the ELISpot or any DOT-related product, which we then capture and analyze, and the consumables. We have a booth, so everybody is welcome to come and talk to me about our products and services if they want to. And with that one, I go to my presentation. As all of you are currently interested and face the challenges of measuring cell-mediated immune responses in your studies. You all want to go from the preclinical phase into the clinical phase. To do that, you need functional measurements because you want to go forward. These need to be there in sufficient quantity and quality. And they need to be effectively monitoring the responses induced. These are challenges because, first of all, they need to be physiological relevant because many things can work great in an academic setting, but once you go out in an outbred population, it needs to be detecting low specific responses. It needs to be validatable. It needs to be objective. So you have to face all these challenges to not just stay in the preclinical arena, but to go forward. Furthermore, to be doable, it should perform with frozen cells. If you cannot freeze the cells, you cannot do like in academic setting, testing every sample individually on fresh samples, and it gets into a very difficult project to perform. So you should have a marker and an assay setup which performs identical with frozen cells so that you can transfer it. Again, it should not just work with a very small specific population with a certain HLA type, because again, you will restrict your responses. And you should have sufficient materials available to work with them. So what type of assays are out there which most of you are very familiar with and work with? ELISA, Luminex, cytokine beta rays, ELISpot assays, flow cytometer measurements where you look at phenotyping by surface marker staining, tetramer, pentamer. So what is inherent in these assays? Every time you choose an assay, you need to realize what are you choosing at. Is it a liquid-based assay, a supernatant-based assay, which measures already released products, so no cells are involved anymore? Or are they real cellular assays, meaning you need functional cells for the readout? And this is guiding you in the selection process and understanding what you have selected. As more cytokines will not increase your sensitivity of the assay. It increases the analytes you analyze, but not how sensitive your assay is. With that one, let's look at this. Which assays are how sensitive in the measurement of the released product of your interest? We did a very simple experiment, and we repeated this with cell lines, clones, primary cells. The principle is very straightforward. You, for example, take CHO transfected cells, mix them with CHO untransfected cells with a certain product of your interest. Mix the transfected cell in a ratio so that you always have 1 million cells in total, and then measure the product of your interest with different assays. So we used the flow cytometry first, so you always have 1 million cells. Then you measure with intracellular cytokine staining the products, and you titrate it down. You see a very nice linear correlation as you go down with the cell numbers of the transfected cells until you reach about 1,000 producing cells in a million. Then you come in the background noise. You might be a little bit tricking it and maybe very hardly push it down to 500, but then you are really at the borderline, really. So you need at least 500 producing cells, or better 1,000 in a million of a certain type to be able to detect it. If you go with ELISA, you do the same thing. Culture the cells, collect the supernatants, then assay them with multiplex, ELISA, whatever you want. Beautiful titration until you come in about 1,000 producing cells. Then you are at the lowest OD levels. Again, you cannot, that's what it is. ELISpot. You have over 1,000 cells, it's too numerous to count. And you get a beautiful linear correlation if your frequencies are lower than 1,000 than in a million, which says that all these assays have the same specificity, but different sensitivities. So selection of assays needed for your readouts are crucial, as if your assay is not sensitive enough, you will not be able to measure the responses. Furthermore, it shows you that you have linear correlation. It can be directly ex vivo measured. So I will focus in my presentation on looking at the T cell and B cell responses in adjuvants on the ELISpot and its technique. So what is the advantages? It is very sensitive. It is directly ex vivo measurements will be measured. No HLA typing needed, so diverse populations can be measured. And no pharmacology treatment of the cells is needed. So no culture, no expansion phase. You can go directly ex vivo and make measurements. Okay. So I told you you need to be able to measure fresh and frozen the same way. This is now basics to be able to do measurements, and here we have very nicely shown that different cytokines, antigen specifically, with interferon gamma, IL-2, IL-4, IL-5, behave fresh and frozen identically. They are plotted, and calculated regression curves led to linear correlations. So we have over and over one of the validation parameters that this assay performs linearly. And fresh and frozen cells behave the same if handled correctly, as these are functional cells. So it is validated and capable. It performs linearly and has a single cell resolution, so high sensitivity. Most of you, I think, are familiar with it, so I do not want to bother you about the basic principles, what ELISpot is and why the sensitivity of the assay is so different than like with ELISA and flow cytometries. All of you are familiar that T cells actually go into the simplified diagram that you have first naive cells, then as they are primed in the surrounding of third signals, which can be adjuvants, solutions, cytokines, inflammation, whatever it is, the naive cells will be primed and accordingly driven to a Th1 type or Th2 type of immune responses, where diverse T cells with diverse cytokine profiles, however, are producing diverse cytokines and immunoglobulins. Most people only measure total IgG. They do not really look at the subclasses of IgGs. But the subclasses as well in the B cell level will show you what type of immune responses were guided and produced. Okay, we don't need to do the basics. I think we have heard about this plentiful, how adjuvants work and what their main contribution is in T-cell response measurements and antigen-induced immune responses. However, I wanted to point out that many adjuvants behave the same in classical readouts, like, for example, proliferation. In proliferation, it looks like all adjuvants, independent on the location they're injected to, they give you a good proliferative response. Why? Proliferation is IL-2 based, so until IL-2 production is seen, you get proliferation. Doesn't matter what adjuvant and what location you are injecting to. However, only the stimulation index will vary, so we will get a stronger or weaker response. If you look at the total IgG responses at diverse adjuvants injection, here again, you see similar capabilities, similar induction of immunoglobulin levels after single injection on the locations. However, here, if you look at the subclasses, there suddenly the population starts to split up. So you see that aluminium induces a mainly IgG1 of B cell responses, versus CFA induces an IgG2A response as well, beside the IgG1, and only CFA does from the tested adjuvants here. If we now look at the T cell level, and here now I will focus first on the CD4 T cells and look at the cytokine differentiation and production after immunization. And locations. What you see here is that in IFA immunizations, you have a strong IL-5 response. So the light dots are IL-5 producing LA spots, and the dark spots are interferon-gamma producing cells. So what you see, the location in IFA defines the strength of the immune response. However, it is mainly Th2 driven by the production of IL-5. If you look at CFA with the same antigen, the only difference is the adjuvant, you actually can switch the immune response and produce a lot of interferon-gamma. And this is independent from the genetic background. Aluminium is a very weak T-cell immunogen and has a stronger IL-5 inducing immune T-cell response than interferon-gamma, independent from the location, so subcutaneous is almost nothing, and with aluminium-induced responses you get an IL-5 response, and soluble PBS doesn't do anything. So this alone shows you already that we have now 3 different results depending what we are looking at. Is it proliferation? Is it IgG production? Is it T-cell responses? Furthermore, you can say, oh, the antigen, it matters if it's a parasite or a protein or peptide. Whatever we looked at, it didn't matter. It overrode. The adjuvant overrode all the intrinsic characteristics, and the same was seen. So for Leishmania, if you gave it in IFA, then you had IL-5 production, CFA, interferon-gamma. Again, independent of the strain. It went autoimmune, MBP, the same thing. So the adjuvant overrides the characteristic of an antigen, if there is. Furthermore, is the age critical? So looking at, in contrast to the data my colleague showed just previously, where it played a role, which age it was given, here when we looked at CFA and IFA, the age did not play a role. Yes? So here IFA induced an IL-5 response, and CFA induced an interferon-gamma response, independent if it was given in neonates or in adults. Okay, you say, good, but who says that the same epitopes are recognized, right? Because still we have a whole protein, we are focused on CD4 responses, it might be that different epitopes are dominated. Now we did epitope mapping and looked at the epitopes recognized and responded to. And what we saw is, again, it was exactly the same peptide, so the same immune dominance of the individual determined were maintained. The only difference was that they produced different cytokines depending on the adjuvant given. Complete switch. And that was very surprising. So now let's go into tumor immunology, where the frequencies of T-cell responses are low. So again, as well, my previous colleague did an introduction on tumors. One thing I want to add here, again, that we should keep in mind the diverse pathways in which tumors can be recognized. Is it a direct pathway or is it an indirect pathway? Because accordingly, different population of CD4, CD8 cells will respond. Like in the direct pathway, CD8 cells will need to respond to the tumors directly on the peptides the tumor express. Or is it that the tumors are absorbed by dendritic cells, then digested and presented, in which case CD4 cells and CD8 cells will respond to? So a little bit of the path we need to keep in mind. And to understand what will happen, the question was, do tumors itself actually induce an immune response from the get-go? And how do we influence it when we expose them to antigens and treatments with vaccination processes? So diverse tumors were used, diverse adjuvants were used, and accordingly, just looking now at the naive mice now, and looking at tumor responses, what we found was that all mice had immune responses against the tumor already. However, they were all Th2 type with low IL-2, a lot of IL-4, a lot, and very little IL-5. And IL-4 is actually priming, is a third signal, which drives the Th2 response. So the tumor guides its immune response to a way it wants it to be. Good. Now, if you look at a clone of LR, which is a tumorigenic tumor, and we used LS, which is a non-tumorigenic tumor, what we saw is the only difference here was the non-tumorigenic tumor actually produced interferon-gamma instead of IL-4. Now, what responses is this CD4? CD8 driven, we clearly showed that the IL-4 was produced by CD4 cells, and in the case you were protected, and to the non-tumorigenic you had a CD8 component as well. Okay. Now, if you provide the cellulars, also the non-tumorigenic tumor, first into the mice and inject it, and then challenge those mice with the tumorigenic tumor, will you protect the mice? So cellular treatment. And yes, we got complete protection from the tumor against tumor challenge, just by the fact by giving them, pre-injecting them with cellular components of the non-tumorigenic tumor. What cytokines were induced? Again, now we got both interferon-gamma responses and maintained IL-4 and 5 responses, but the frequency of the interferon-gamma responses were dramatically increased, and these responses were CD4 and CD8 driven. So we can say we added now looking at peptides, because these were all complex, whole tumors, cellular products, proteins. What about peptides? Now, going into peptides, we had shown that actually CpG is a very big adjuvant and induces a strong interferon-gamma response. And now we then studied that in tumor models and used peptides, individual peptides with the diverse adjuvant, and looked if the peptide directly induces a response against the tumor. Here what we saw, that we had a strong adjuvant effect of CFA, which was antigen independent by producing IL-6 and TNF, and there was a hierarchy between the adjuvants used. Some adjuvants were much stronger in interferon-gamma production, and some had more stronger responses in inducing actually IL-4, IL-5 responses. So there were diverse cytokines induced in a clonic environment only by using different solutions, if you wish, or adjuvants, when you injected this into the animals. Now, what is protective? You know, it's all nice that we have the cytokine profile, but now what about the protection? What we actually see that the adjuvant effect actually was dramatic. It increased the livelihood of the mice from 20 to 30 days. Just the adjuvant itself, without any antigen specificity. However, if you added the CpG with the strong interferon gamma, you got a third of the mice which were completely protected, and this could be transferred, and it was an and hierarchy was important. And I want to point out this hierarchy because you will be surprised to see later on that CD8 cells, this hierarchy is not the same. So CD4 cells and CD8 cells behave differently to the same adjuvants. And now depending on, and I want that you keep this in mind, that for CD4 cells, CpG was the strongest interferon gamma producer, and CFE was not too good in producing interferon gamma. And you could transfer this. So this is all nice. You can protect mice before they see the tumor, but that's not what happens in humans, right? You need to have a tumor. You have already a tumor, and then you want to treat the individuals. So now if we injected in a growing tumor, this is size of the tumor, we injected CpG with no antigen, just CpG itself, because the tumor, the idea behind this is that the tumor itself provides all antigen needed. T cells are there. They produce the wrong cytokine, IL-4, right? So what happens if you change the milieu and give them CpG? So we needed to inject 3 times, and you saw that the tumor regressed and never grew out again. So actually the mice were cured from the tumor itself. And what was their cytokine response? What we saw now again, that we got a very strong interferon gamma response after the tumor injection. And this interferon gamma response was very split between CD4 and CD8 T cells. Yes? Just by changing the environment in which the tumor is recognized can induce an anti-tumor specific response where we don't know what the epitope is, what the response is, but it responds to the whole tumor itself. I call that determinant spreading. So we induced huge determinant spreading. This could be transferred, the other animals protected. So what about CD8 cells? Because, one more important thing was that as we saw, and look now in autoimmune diseases, we saw that the adjuvant effects are different for CFE and CpG in IL-17 production, as CFE induces a very strong IL-17, whereas CpG does not. Consequences in autoimmunity are drastic. CFE induces strong inflammation, whereas CpG does not. No DTH. Disease induction only by CFE. CpG does not. And if you treat animals which got IFE with anti-IL-17, you could help the animals to recover. Which means is the adjuvant CFE CpG produce the same amount of interferon gamma and IL-2. However, only CFE produces IL-17, induces DTH, and as well disease. So I don't know now how this is affecting and translating in other adjuvants, but I think we need always to look at multiple cytokines and multiple readouts to understand the impact of the adjuvants which is there on the T cell responses and their consequences. These were CD4 cells. Now looking at CD8 cells. Now we used peptides, clean defined antigens. Here we saw that now actually the peptides, the CpG induced stronger interferon gamma responses than CFE. CFE, actually, yes. Yes. And then, that's mixed up. And only CFE induced IL-17 still. When we looked at killing, however, the cytolytic responses from CpG were very strong, so that was in vivo killing in mice, and which was only seen in CpG, and CFA itself did not induce peptide-specific killing in the animals. So we got a diverse portfolio here. For CD8 cells, the CFA interferon-gamma response was the strongest, CpG was lower. In CD4 cells, we had it the opposite way. CpG induced a stronger interferon-gamma than CFA did. The same was true for IL-2, so we have flip-flopped now the strength of the immune response from CD4 to CD8, depending on the antigen usage and targeted. IL-17 was still induced by CFA only. Now cytotoxicity was split. CpG induced very strong cytotoxicity and no DTH, whereas CFA induced no or very little cytotoxicity and strong DTH. So I think taking this into consideration in adjuvant selection is an important process, and this is as well reflected in the population measured and the kinetics of times. And then I wanted to look at the B-cell responses and the diverse B cells, meaning now of the spontaneous antibody-secreting cells in the blood. Once after treatment, they migrate from the lymph node to the bone marrow, and you look and directly measure them in the blood, how they respond and what the benefits of them are. And here we look now at diverse isotypes in them in a 4-color B-cell ELISpot assay. With that one, we could classify the 4 different isotypes in one well, and then look at antigen-specific responses. Here a double color, here multicolor to multiple cytokines. Is this relevant in the human? Looking at CMV responses. In humans, you see a very nice distribution of the 3 different immune globulins dominated by IgG, some IgM and IgA responses in the blood directly. Is it the same in every individual? So now we have 1, 2, and sorry, this is missing 8 individuals. And you see the distribution of the immune globulins in the blood is very different, also the plasma B cells, yes? And they produce different amount of the immune globulin. So what is protective, what is not, how strong the response need to be, that are all questions which I cannot answer, I don't know. And I don't know if you can answer. And of course, other application processes are there. And with this one, it is validatable, the assay. So we had worked with different labs throughout the country, provided a lot of reagents and solutions to standardize the whole process. We served as reference laboratory, provided the responses, compared the responses between the diverse labs, and high responder, medium, low responder were all repeated and reproduced easily, and where the dotted line is the 3-fold standard deviation. And accordingly, it shows this is a very robust assay, can be produced. Very important is to work serum-free, as serum alone is an antigen and produces huge responses in individuals. With that one, I would like to thank the people who actually worked with me on this project in my academic settings, and I will take any questions you might have. Thank you very much for your attention.
Thank you for that talk. We have time for one quick question. So I think it was a nice demonstration of that we need to dig deeper, and that, you know, we've looked at vaccines very superficially when we just measure a neutralizing antibody and we don't care about its isotypes or what's happening to the CD4 or CD8 T cell responses.
Correct.
But on the other side, of course, to dig deeper is an enormous amount of work and quite expensive. Do you have any comments on that? I mean, how do we build this into our clinical trials without pushing their costs from, you know, hundreds of millions to billions?
Very simple.
Because this is expensive work.
Correct. I think it is a good idea to start preclinical, to look in this one in mouse models, because there the volunteers are easily recruited. And this should be dissected early on. We get requests in phase 3 to start looking at that. Of course, if you start in phase 3 looking at T-cell responses and then start to establish an assay for your clinical trials, then it is very costly. So I think as earlier people do it, the better it is. However, it is interesting to see that people have a very big resistance against looking at T-cell responses and going away from the total IgG responses in vaccinology, because these assays are allowed for so much longer, so the confidence is there to go what they did all the time, even though if their vaccine actually targets T cells. So I think it should be a choice of the purpose why the vaccine is induced and what type of immune responses you want to induce, and do it as early as possible to reduce your cost and not wait until you are forced by the FDA to do so. That is
Thank you very much. So we'll move on to our next